Hi everyone, this is Dr. Hamza Anwar, Head of Research at the Cure Brain Cancer Foundation. In this video, I'll be recapping some key findings from the 2024 European Association of Neuro-Oncology meeting, which took place in Glasgow a few weeks ago. Day 1 opened with an important session on patient and public involvement in brain tumor research and highlighted the increasing role of patients' voices in the research process. One talk shared results from a phase 1 trial that captured patient feedback on proton therapy for glioma and GBM. Patients noted that participating was not only empowering, but also contributed to a sense of personal growth. Later in the day, the focus shifted to DNA repair inhibitors in glioma treatment. Presenters explored the latest advances in DNA damage response inhibitors, which prevent cancer cells from repairing themselves post-therapy. New updates included studies on PARP inhibitors like olaparib, which when combined with radiotherapy, led to over 2 years of survival in 25% of newly diagnosed GBM patients, and niraparib, which showed a median survival of 20 months and also reaching higher drug levels in GBM tissue than other PARP inhibitors. Day 1 concluded with an update on IDH inhibitors and their role in treating brain tumors. This included findings from the INDIGO trial, which showed that vorasidenib, an IDH inhibitor, slowed tumor growth in low-grade gliomas with IDH mutations. Remarkably, the treatment had minimal side effects, with patients experiencing no noticeable decline in quality of life. The success of vorasidenib led to its FDA approval in the US and TGA approval in Australia for treating low-grade gliomas. Day 2 opened with presentations on democratizing access to clinical data. Presenters discussed how secondary use of patient data could advance personalized cancer care. In particular, medical imaging offers valuable insights through imaging biomarkers, but much of this data remains underutilized. Next, talks focused on new targeted therapies for gliomas. Talks explored ATR inhibitors, which can reduce tumor burden and limit GBM invasion when paired with traditional treatments. These inhibitors have shown potential for making tumor cells more sensitive to therapy, particularly in cases where resistance has been an issue. Another session focused on targeting TERT, a gene linked to cellular immortality in tumors. Day 3 wrapped up the conference with talks that focus on navigating preclinical approaches to identify windows of opportunity and develop clinical trials. One talk discussed how to navigate the preclinical to clinical pathway, with case studies of researchers finding themselves in a state of limbo due to failed preclinical work, but ultimately thinking outside the box to restart the engine and get their work back on track for clinical trials. In another talk, researchers presented a patient-derived GBM drug screening platform, which selects drugs based on their ability to penetrate the brain and work effectively in GBM cell cultures. These drugs will be evaluated in a window of opportunity trial to test their effectiveness, with promising candidates progressing to clinical trials for personalized treatment options. In conclusion, the 2024 EANA meeting was a tremendous success, providing a platform for knowledge sharing and collaboration in neuro-oncology. The Cure Brain Cancer Foundation was proud to be in attendance, gathering insights that will help refine our strategies and strengthen our commitment to the brain cancer community.